maybe I can first share my screen with you. So I'm Gülşah and I am interested in human population genetics. I received my PhD from Bilkent University from the Department of Molecular Biology and Genetics. Then I proceeded to the postdoctoral studies, and during the postdoc, I worked with ancient human genomes to study the past human population dynamics. Currently, I am affiliated at the Hacettepe University at Department of Bioinformatics, and now my focus is studying the dynamics of evolutionary forces that are shaping the genetic variation behind the complex human diseases. Today, we will be together to learn about the forward time genetic simulations. I feel a bit unlucky because this is one of the latest courses of the program. You can be a bit tired, but I will try to not to cover too many subjects, but the course will be like giving you a basic idea about what are the simulations, why do we need them. Actually, yesterday, Pavlos also mentioned about the coalescence simulations. So today, I will just mention about what are the basic approaches that we run the simulations, the forward time and backward time approaches. And then the course will be about learning one of the most famous softwares to run the forward time genetic simulations, which is SLiM. There are so many materials about it on the web. You can find it. The manual is very open and clear. When you read it, you will recognize it. And also there are some workshops that are organized by the developers of the SLiM. So today I will tell you about what is a SLiM and what kind of scenarios can we simulate with this software, and why do we need the forward time genetic simulations. So the course will be like this, and at the end, maybe if we have time in the last course, we can just try to write a simple simulation scenario together without running it, but you can just try to run it later on your own computers after the course. So let's start. So this is the plan of today's lecture. At first, I would like to tell you about why do we need simulations in population genetics and what are the basic approaches, as I said, and what parameters should we consider while designing the simulation study. So we decide to run a simulation study, but while designing our simulation, what should we consider regarding the population properties? And then we will learn about what is SLiM, and we will follow the manual of the SLiM a little bit. And then we will create a simple simulation scenario together. So I always start from the Big Bang. The variation, as we know, is the source of the life. And this variation in our lives is explained by the genetic variation. So the population genetics is the discipline that we study the genetic variation in different populations, in different species. And when we say genetic variation, what we study is that here, if we say that this is the DNA sequence, which is composed of 4 different nucleotides, and then any change of this nucleotide sequence is called as variation. And this makes our lives. And we study the evolutionary forces and also the demographic dynamics that shape this kind of genetic variation to understand the basics of this. So we can think that the individuals and also the populations and species, they are all shaped by complex processes. There can be some random mutations, or there can be some demographic changes like population expansion, or diseases can generate some selective pressures on the population. There can be admixture between different populations or gene flow from one population to another. The populations can also experience some bottlenecks, and also there can be genetic drift. So all these complex processes, all these evolutionary processes shape the genetic variation that we see in the whole population. And the thing that we do in the population genetics is to study this genetic variation. Why do we do this? Maybe I can give you an example about it. So this is a principal component analysis result, and this is an example from the human populations. But you can think, you can generalize it in your mind for different species. So if we look at this figure, this is a principal component analysis that is summarizing the genetic data of the individuals into two dimensions. Here, each dot corresponds to one single individual. For example, here we see that the green individuals here, orange individuals are here, and then the red ones, green ones, each color represents one single population. And the color codes are here. You can see them here. For example, here we see that the Near Eastern populations like this, and then some Armenian, Iranian, and Turkish populations here. And for example, here we have some South European populations and West European populations, for example. Then what we see here is that there is a pattern that is consistent with the geography, and individuals are grouped together based on genetic material. This analysis is done by running the principal component analysis on a set of SNPs, more than 500,000 single nucleotide variations. And then the individuals are summarized based on their genetic variants. And then you see this pattern. And this pattern that we see here is shaped by multiple processes, like the populations migrate at some point in our prehistory. So these are the modern populations that live today. But all these patterns are shaped by the past processes that the human populations in our prehistory experienced. For example, they migrated at some point from one part of the world to the other, or after they migrate at another time point, they admixed with each other. And such kind of processes, evolutionary processes, make this genetic variation. And this all these genetic variation is shaped by the evolutionary forces and the demographic changes. So we see that, for example, if there are different populations, so between different populations, there can be a gene flow, which means that the population can migrate from one place to other, and there can be a gene flow. Or there are some selection processes that change the trajectories of the allele frequencies towards another point. Also, the effective population size of the populations can change, which is importantly affect the structure of the population. In some cases, for example, there can be migration from one place to another. Or in some cases, there can be admixture. You can think that all these dots can be alleles, and when there is admixture, the alleles are admixed between populations. In some cases, after admixture, then the population which is admixed can be expanded, and then all these processes change the allele frequencies. And we can track these allele frequencies to understand the patterns of evolutionary forces, and to understand how do we have this kind of genetic variation today. To do that, one of the approaches is to use a time series data. What I mean is that you have a population and get the samples from different time points, time point 1, 2, 3, 4, and like 100. And then sequence the DNA, get the genetic variation, and follow the pattern of the genetic variation. This is one way. And this is possible due to improvements of the ancient DNA methodologies. Now we can sequence the ancient specimens that are coming from thousands of years ago, and then we can follow the changes in the allele frequencies in time. Another approach can be producing the pseudo data, like that we call as in silico data sets, that is covering the genetic variants. And to do that, to produce the genetic data, there are, like in silico genetic data, there are different approaches, like backward approaches or forward time approaches that we will cover today, and some resampling based approaches. And while doing this, while producing the in silico data sets, in silico genetic data sets, we consider a set of scenarios that cover different evolutionary forces and different demographic changes. So even if we have real genetic data by sequencing the ancient genomes and modern genomes, the simulations are good in terms of providing a perfect environment. Maybe we should not call it as perfect, but we can say that you generate genetic data under conditions that you know. So you generate migration, and then under this migration model, what kind of genetic variation pattern you see. Or you produce different populations with different demographic histories. For example, one population is starting with a low population size, and then effective population size increases in time, and then other population, and admix them, get the samples, examine the DNA sequences, and then you will see that everything is clear. So you have the scenario, and then what is the expectation based on this scenario? Because of that, the simulations are really good tools for us to study the population genetics and evolutionary forces, dynamics of the evolutionary forces that shape the genetic variation. While designing a simulation study, what we do is to decide how to choose which program or which approach should we use. I also forgot to say at the beginning, but if you have any questions while I'm talking, you can just interrupt me and ask your questions anytime about the thing that I'm telling you. So when we are designing a simulation study, we should first need to know about which approach should we use. So there are forward time approaches and backward time approaches, but what should I choose? What program also should I choose? Because there are many different programs that are running forward time and also the backward time simulations. So this depends on the aim of your study. And because of that, we need to first decide about the aim of the simulation. So I will run a simulation, decide to run a simulation study, but what is the aim of my simulation study? There have been 3 different purposes. In general, we can generalize in that manner. So you can also read this Hobon 2012 paper. It is a bit earlier compared to 2021, but it is a very comprehensive paper that gives you an idea about how to design a simulation study and how to select the appropriate simulator for your study. So the first thing that we decide is the aim of the simulation. One purpose can be the predictive use. So when we say then I'm at home and I don't know what to do now. Maybe I can just ignore. Okay. The predictive use is like, so when our aim is to use the simulation as a predictive purpose, then it covers the theoretical studies. For example, if you want to study the sexual reproduction dynamics, or if you would like to study the speciation, then you can, maybe I need to, sorry, open the door, sorry.
Sure, there's no problem.
Unlucky aspects of studying from home during the pandemics, but it's fine. Let's share again. Sorry again. So the one purpose was the predictive use. Then we are doing this in a theoretical purpose if you would like to study the sexual reproduction or speciation, for example. Or if we would like to study the conservation, for example, so imagine that there is a species that needs to be considered for conservation. Here, the simulations can be used to predict what approach could be better to conserve that species. So at this point, we can also run the simulation studies. I will show you one example for that in the coming slides. And also we can study the epidemiology for the predictive purpose. For example, like in the complex diseases, there is common disease, common variant hypothesis like this. And so to investigate if this hypothesis is correct or not, so what are the dynamics, what are the expectations from a population under the common disease, common variant hypothesis, we can run the simulations with the predictive use. Another approach can be like statistical inference. Then what we do is like, if we would like to study the evolutionary history of a species or demographic changes or migration patterns in single population or between populations, or if we would like to study, for example, what was the size and duration of the bottlenecks that the population experienced, we can also study the strength of the selection at particular genetic loci by simulating the genetic data again. And also we can, for example, study the divergence of the species and the populations, as well as like kinship coefficients in both plant and animal species. So if we would like to study all the dynamics of these processes, then our aim is like statistical inference. For that purpose, we can also use the simulations. And there is some other purpose that in population genetics, we don't use that much, but maybe in medical genetics more appropriate. Then you can just do the power of assignment test or power of genome scanning methods. So you can use the simulations to validate the efficiency of the statistical methods. So in population genetics, we also use it to validate the efficiency of the method that we developed, for example. So after deciding the simulation study's aim, then what we do is to define the scenario and the parameters. So while defining the scenario, if our aim was a predictive, if we have a predictive purpose, then while defining the scenario, we need to consider like the theoretical questions. So the question can be, if you study the sexual reproduction, for example, then your question might be like how sexual forms have evolved. Or if your aim was like epidemiology study, then you can ask a question about like what the basics of the common disease and common variant hypothesis. Or you can ask a question like, can historical bottlenecks produce excess of damaging alleles in population like we see in the African, out of Africa populations. And if our aim is like studying the evolutionary dynamics, and if our aim was to get a statistical inference from the data, then we need to have a prior knowledge on the competing hypotheses. Like imagine that there is a scenario that is acting on an allele, for example, like the strength of selection on particular loci. So the competing hypothesis might be what was the patterns of the selection. And also, while our aim was validating the statistical methods, we can just consider the common use of the proposed methods in the field. So these knowledges are important for defining the scenario for the simulation, and then the parameters. So we decide the aim first, and then define the scenario. And what we do is, deciding the statistical tests that we are going to run on the simulated data. For example, this is an example from Bruford 2010. Here they use the simulations for predictive use, as I said, like here for the conservation purposes. Here there are orangutans that are living in different islands, and then due to low population connectivity between these orangutans, the thing is that there is inbreeding within each population. And because of that, there is a population decline of the there's a decline in their population. So the aim of the authors was like producing a management strategy to conserve the species. And here the management strategy that they defined in their study was, one of them was no intervention, so doing nothing for for conserving them, or just establishing some corridors like migration corridors that will allow species to migrate from different places, or translocation. And then the other approach was like combining both, like both corridors and translocation. So the question of the study, for example, here is, which approach is the best approach to conserve this species? To understand that, you can run the simulations. And while running the simulations, you will test something. So you need to decide the statistical test. For example, in that case, what kind of statistical tests that they decide was probability of extinction. So if you do no intervention, what will be the probability of extinction? Or if your approach was making like setting corridors, then what is the probability of extinction? And also they evaluated the final population size after applying all these approaches, or what is the expected heterozygosity, inbreeding coefficient, or level of genetic differentiation. So as you can understand from this part, so the statistical tests that we run on the simulated data depends on the purpose of the study. So since here the aim is finding a management strategy, they simulate scenarios without intervention, or simulate another scenario with establishing migration corridors, translocation, and combining corridor and translocation. Under each scenario, consider some parameters like population-specific carrying capacities or translocation rates, and using these parameters, simulate different scenarios and run the statistical tests to understand what is the best way of doing management on this population. And after deciding the statistical test, we also need to select a simulator. So there are different simulators, both backward and forward time. There are many different simulators. Everyone is developing their own simulator in some cases. And we need to decide it depending on the capabilities of the simulator, because some simulators, for example, I will mention a little bit later, some of them do not consider the population. For example, some of them cannot incorporate the selection. Some of them are not efficient in allowing the different mating systems. So depending on the capabilities of the simulation software, you can select it. And then after selecting in a normal simulation study, what we do is running the simulations. And before running the simulations, what you need to do is deciding number of replicate runs. If you're running a coalescent-based simulation, which is backward time simulation, or if you're running forward time simulation, then you need to decide about the number of generations per run. How many generations are you going to produce in each run of the simulation? So after deciding this, we run the simulations. And after running the simulations, we calculate the summary statistics, and then we get we reach to a conclusion. For example, in that study, their conclusion was like to protect this species, both combining the corridors like migration corridors and translocation was the best way. So they decided that. Another example can be here. So what we do here is that, so this study is from Talman et al. From 2006. And this is about studying the evolutionary and demographic history of the gorillas. Here there are different gorilla populations. And then they are studying the demographic and evolutionary history of this population. For that, in such example, for example, what they are doing is deciding first alternative models. For example, you can decide if you study the dynamics of the evolutionary forces and the demographic changes, then you need to create different models based on different effective population sizes and different divergence times. So then they tested if after the split between east and west side of the gorillas that they study from the Africa, no migration or migration was an equal amount between the west and east, or asymmetrical migration. So the scenarios that they simulated and tested was like no migration between east and west gorillas, equal migration or asymmetrical migration, which means that west is migrating to east more or east is migrating to west more. And then they consider the timing of the split between the population and population size estimates. In such case, for example, the statistical test, the summary statistics that they use is diversity estimates and also studying like the differentiation, like divergence between the populations, the genetic distance between the populations using some FST based or Fu and Lee tests. They investigated their question. And as a result, in that study, for example, when they run the simulations under this scenario, what was their condition? What was their conclusion? It was there was more gene flow from eastern to western gorilla population than vice versa. So there was an asymmetrical migration. So the simulations are good in both cases, like in studying the evolutionary dynamics, in studying the conservation or epidemiology. So you generate an in silico data set, then study the forces that is shaping this genetic variation. While designing the simulations, what we need to do is like deciding the parameters that we need to consider before selecting the simulation approach. So since today we are going to meet with SLiM and forward time genetic simulators, first question coming to mind is that why do we run the forward time instead of the backward time simulations? We need to know about their basics, what kind of data that you can generate between different approaches. The both of them has some pros and cons regarding different patterns, but the parameters that we need to consider, one of them is the life history, which can be characterized by the life cycle of the species or the population, and the mating system or hermaphrodites or like the reciprocal mating, and age structure of the population or the sex system in the population. And while considering the life history, if you're going to simulate a basic life history, then the backward approaches, the coalescent-based approaches are better because they focus on just genes, not the individuals or not the populations. You simulate the genes, which is very simple, which is very straightforward, and which is very efficient. But if you would like to generate like complex life history scenarios, then we mostly rely on the forward time approaches because they can incorporate different parameters to the simulation. So for example, when you say life cycle of the species, there can be a complex life cycle with multiple overlapping stages. So like in the case of the long-lived plants or animals. So the stages of the population can interact, can overlap in each cycle. But on the other hand, there can be also two stages based on the reproductivity of the population. And also mating system can be different. Like there can be some populations that are hermaphroditic when there is random mating and self-fertilization. In some populations, you see that there is polygamy or monogamy, or you can even see that phenotype-based assortative mating. So if there is such a complex mating system, then it is not very feasible to use the backward time approaches. The forward time approach is better here. And also you need to consider about the fecundity and population growth parameters while deciding this. So the basic idea here is that if you would like to simulate a complex life history, then go for the forward time simulators. SLiM is not necessary, but you can select another forward time simulator. But I will tell you about what are the advantages of the SLiM over the other simulators. Another parameter that we need to consider is the migration. Here, what we do is like if there is in the basic scenario, you can define a matrix of the migration rates. And for example, if there is a rice island model, that there is migration from one island to another, or there can be like complex migration pattern that is spatially explicit, or in that case, the migration can be affected by like the environment effects. And in some simulators, there isn't any opportunity to incorporate the migration. So if you would like to study the migration, then you need to go for the simulator that has an option about incorporating the migration. So these patterns are the ones that you need to consider before deciding the simulator. So you read the manual and then decide if you would like to simulate the migration, and if the simulator that you go for it doesn't have any capability of doing that, then you change the option. Another parameter that you need to consider is recombination and mutation. And in that case, the simulators, some of the simulators have, all the simulators indeed allow the mutation. So even if you use the backward or forward time simulator, all of them allow the mutations. And all of them use various mutation models. And mutation rate, for example, when you are running the simulation, it can be constant, or you can get it from a gamma distribution, or in some simulators, for example, the recombination is allowed, and it can be constant across all the sites. So if you simulate, for example, 10 nucleotide long sequence, then this is very like simply example, but so it is constant across all the sites, or it can vary between different parts of the chromosome, for example. And in some other simulators, some of them allow selection and phenotypes, some of them not. And mostly the forward time simulators allow this selection. There are some coalescent-based methods that also incorporate the selection, but the forward time ones are more efficient in simulating the complex selection scenarios. For example, if you would like to study the polygenic selection, there is no way to do this in a coalescent simulator, as I know. At this point, while considering all these parameters like lifestyle, migration, recombination, selection, then we have different simulation approaches, as I said from the beginning, like backward time. This is a coalescent-based approach that you learned yesterday. And there's forward time approach and resampling approach. So the backward time approach, you already learned about it. It constructs genealogies rooted in the most recent common ancestor. And the advantage here is that with the coalescent-based approach, you can simulate extremely long sequences, but the difficulty here is that you cannot handle the complex scenarios, like you cannot produce the diploid individuals, or you cannot simulate like complex polygenic selection patterns on multiple sites. But they're computationally fast. So depending on your question of interest, you can run the backward time approach. In the forward time approach, instead of the genomes, like sequences, now you focus on the individuals. With the forward time approach, you can simulate the individuals. And it starts from the past, and then it comes in time to the current day. And you can then track the entire ancestral information, and you can follow the changes in the genetic structure by analyzing individuals by drawing them at any time point from the simulation. So simulate the data, start from time 10,000 years ago, and then come to the present day. And you can just sample individuals, for example, from 5,000 years ago, 6,000 years ago, 2,000 years ago. So you can get like samples, individuals from each time point. But the difficulty here about the forward time approach is that they are computationally more demanding. So they require more RAM, and so they are not very fast as you do in the coalescent-based approaches. But the good thing here is that you can deal with the complex evolutionary scenarios by using the forward time simulators. There is also another approach that we don't use that much, but it's also very useful for studying maybe in the epidemiology studies. This is the resampling approach. In resampling approach, what you do is bootstrapping from an existing data set like 1,000 genomes or HapMap or any published data set. And they are even faster than coalescent and forward time approaches, but they are not good for generating the genetic diversity. So you cannot simulate the complex and evolutionary scenarios. But through resampling approach, by using an already available genomic data set, then you can simulate unlimited sample size with case control or affected child trio. So it's better for studying the diseases, for example, or for the GWAS studies. This approach is better. So what we have is that mainly, I'm not going to cover the resampling, but I just put this, so you learned it yesterday, but I just put it for comparison with the forward time approach. In the backward time approach, Here, imagine that this is the current generation. You start here and go back in time until you coalesce all the samples into the most recent common ancestor. And then you can track the evolutionary history of the genes. And after generating the tree, you add the mutations. And this kind of mutation, this kind of simulation approaches are mostly implemented based on the Wright-Fisher model that I will tell you. So the Wright-Fisher based, it is a simple population genetic model that assumes that there isn't any selection, there is random mating, and the number of individuals are constant over generations. In that case, after finding the most recent common ancestor, you come forward in time and assign the mutation, migration, and recombination. So the selection is not considered, but in the novel methods that run the coalescent-based approach, there's also a possibility to incorporate the selection to the simulation too. When you're running forward time simulations, what you do is that starting with an initial population. So this is the initial population. Let's say that a human population that is coming from 1.5 million years ago. And then simulate the evolutionary and demographic scenarios. Take the population, expand them, and then apply bottlenecks and get a subpopulation from it and another subpopulation, migration between them. You can do whatever you want with the population after starting from the beginning. So start and then run the evolutionary scenarios, generate the new population, and then you check if the criteria that you want to reach is achieved. If not, then run it again. But if yes, then the final population is in your hands. And the good thing is that you can, as I said, you can just get the samples from any time point when you start from the beginning. But the disadvantage here is that in the forward time simulators, you need an initial genotype. And to use as initial genotype, if the real data is absent, then you can just run a burn-in period. Like in the simulator, you, without generating any output, run the simulation thousands of generations to reach to a constant state of the selection mutation balance. And another approach can be seeding the simulator with samples that you produce by a backward simulator. This is one of the pitfalls of the forward time genetic simulators. Maybe it's good to mention about the restrictions of the backward time approaches because, for example, in backward time approaches, simulating realistic and complex evolutionary models is difficult. And you can just simulate the haploid haploid sequences. So you simulate the haplotypes. If there is like different non-addictive selection or penetrance, then you need to ignore this. And if you would like to study the evolution of a disease, and if you run the coalescent-based method, backward time method, so coalescent means backward. So backward time approaches are the coalescent-based approaches. So if you do this, you can just study one loci under constant selection. And so also you work for definite parameters like low recombination rate and low mutation rate. But when you come to forward time approaches, why we choose them? Because you can track, you can follow the sample's history from past to present. Then you can follow the ancestral information completely. And if your question is about evolutionary processes, instead of the outcome of the evolutionary processes, so if you would like to see the evolutionary processes in action, in time, then you can go for the forward time approaches. And also you can incorporate, for example, changing effective population size. So get the population started, as I said, change it, like expand the population or bottleneck in the population. And also you can incorporate complex patterns of recombination and mating. And it's also more efficient in incorporating the selection. But the disadvantage is that, as I said, it's more flexible. Yes, you can do whatever you want, but they are more demanding compared to the backward time approaches. So here there is a list of different programs. For example, this is from Hobon et al. You can go to the paper and check in detail what are those patterns. So maybe, okay, so this is the paper. So you can go for the paper and compare the capabilities of different simulators. But in general, they are different, for example. So these are the list of the backward simulators. There are many of them, and then more recent ones. This is in 2012. And then they have some like capabilities like spatial coordinates, and then maybe migration or dispersal modes. Again, about the mating system, what different mating systems can be incorporated, and fecundity, life cycle, and the population growth. You can also check if you can incorporate selection, for example, in some of the simulators or not, and what is the patterns of the mutations that you can incorporate in the simulators. So for example, here, this is a very famous MSMS, and it is a combined forward-backward approach, but it works on just single locus. And if you come to this, here we have some forward time simulators, except SLiM. So we will see SLiM later, but there are some famous ones, SFS-Code, CIMIPOP, QuantiNemo, Nemo, or GenomePop. These are all forward time simulators. Here, the thing is that, for example, the SFS-Code is highly flexible because in the forward time simulators, there's always a trade-off between flexibility, efficiency, and ease of use. So if, for example, in this SFS-Code simulator, it's highly flexible, but it's less efficient. In CIMIPOP, it is efficient, but you need to define the evolutionary scenario by writing a script in Python. And if you don't know how to write a script in Python, then you first need to learn about it. So it is not very easy to use for everyone in the biology. Here, the SLiM comes to the scene. This is developed by Ben Heller and Philip Messer. You can go to the website of their lab to get the software and also to get every kind of documentation about their program. There are also forums that you can go and check the questions about SLiM. They also organize very nice workshops that they teach about how to use SLiM. There is also a GitHub page for the SLiM. You can also check this page too. This is another simulator, but there is some population genetic scenarios that you can read about as an example to use in the beginning of your SLiM application. So today we will be learning about this simulator like SLiM and how do we run this. What are the parameters that we consider in SLiM, and what is the basic concepts in running forward time simulations in using SLiM, which means like selection on linked mutations. So this is an evolutionary simulation package. Maybe we can come to this course in the next hour. Yes, we can do that. So do we have time, or now we finished, right? John?
It's up to you. If you like, we can take a break right now.
Okay, then we can give a 15 minutes break, then continue with the SLiM.
Okay.